Hello, everyone. Good morning. Welcome to the MPG Primer. Today, we are excited to have Elizabeth Dorans. She's a fourth-year PhD student in Alcus Price's group in the Department of Epidemiology at Harvard Chan School of Public Health. Before beginning her doctoral studies, she earned her undergraduate degree at Vanderbilt University, where she studied molecular biology, human and organizational development, and music. I just learned she played the viola. Her current research focuses on developing computational methods to link regulatory elements to their target genes using single-cell data. So Elizabeth, take it away. Please feel free to ask questions throughout the talk if you have them. There's 2 mics located in the center of the room. Also, if you raise your hand, I could run over and give you a mic. Thank you.
Like I have a laser pointer going on. Okay, I'll just put that down there. All right. Morning, everybody. I'm Elizabeth. I'm very excited to talk to you all today about this new technology that has the potential to measure biology at a really unprecedented resolution. And I actually want to start by kind of framing the goals of my presentation. As you all might know, the number of single-cell technologies, single-cell data sets, and single-cell analysis methods is huge and rapidly growing. There might even be a few out new by the end of this presentation. So with that in mind, the goal of my talk today is less to dive into the specifics of a particular technology or the math of a specific analysis method, and more so to gain kind of a broad appreciation and intuition for the big technical challenges in the world of single-cell technology and single-cell data analysis. All right. So here is an outline of my presentation today. First, we'll discuss kind of emerging single-cell multiome technologies, starting, of course, by understanding what is single-cell technology, and then more specifically, what is single-cell multiome technology. And hopefully the bulk of the discussion will be on analysis methods, which I imagine are of greatest interest to this audience. We will learn what questions single-cell multiome data can answer. We will discuss the major technical challenges currently in single-cell multiome data analysis. And I'll finish with a sort of in-depth look at one example area of inquiry in single-cell multiome data, which is computationally linking enhancers to their target genes. Okay, so to start, what is single-cell technology? So as I'm sure many of you might be aware, single-cell technology refers to a family of sequencing technologies that measure biology at the level of individual cells. So traditional sequencing technologies, referred to as bulk technologies, make biological measurements that reflect the biology of a group of cells, perhaps from a tissue sample or cell culture. They produce one quantitative readout per sample, reflecting kind of the average profile of biology within that sample. Whereas single-cell technologies produce many quantitative readouts, one per cell, reflecting the biology of that particular cell. So it's perhaps intuitive, but why these days is everyone interested in single-cell data? So a popular metaphor highlighting the benefits of single-cell data is the fruit smoothie versus fruit salad metaphor. So we can think of traditional bulk data as a fruit smoothie, a bunch of different fruits kind of ground up and made homogeneous. And we can think of single-cell data as a fruit salad. All of the different fruits mixed together, but their distinct textures and flavor profiles are still detectable. So if we are interested in learning about some characteristic of a sample of fruits, for example, we can gain a lot more information from a fruit salad than we can from a fruit smoothie. A fruit smoothie might be able to tell us the general characteristics of fruit or of a particular population of fruits in our smoothie, but A fruit salad is going to give us that information for particular fruits and even particular slices of the same fruit. So the same intuition can be applied to a more biological example. Perhaps if we're interested in some molecular feature within a cell, like the expression of some fictional gene A. A traditional bulk sample is going to give us one readout, just an average expression level of gene A across all of the cell types and individual cells in the sample, whereas a single cell sample will produce one readout per cell. And certainly in a real life bulk or single cell data set, there'd be way more than 4 cells. So with that, there'd be way more heterogeneity, perhaps even at the level of a subcell type or cell state. So in this example, we can see how this might be compelling. The bulk readout shows that there is some moderate level of expression of gene A, whereas the single cell readout shows that in certain cells or cell types, there's very high expression of gene A, and in other cells, gene A is very lowly expressed or maybe even not expressed at all. We can imagine if we're interested in understanding the function of gene A, maybe it's associated with a particular disease or something like that. This unlocks a lot more insights and kind of leads to more interesting downstream follow-up hypotheses than the simple bulk readout. So to summarize, single cell data retains cellular heterogeneity. It gives us much higher biological resolution and allows us to ask more specific questions. So what types of biology can we profile at the level of a single cell? Single cell genomic technologies measure a variety of biological modalities, ranging from DNA sequence to gene expression, chromatin accessibility, biochemical modifications to DNA or histones. This figure here just lists a small subset of the biological modalities and corresponding sequencing technologies that are currently available, and is certainly vastly expanding. But these modalities are often uniquely informative and usually provide complementary insights. So for example, if we run a single cell RNA-seq experiment, get a profile of gene expression across a population of cells, we might observe the expression of a particular transcription factor of interest in a subpopulation of cells. And we might hypothesize that this transcription factor is active and is turning on some known gene regulatory network. Perhaps some information that could be very useful for validating this hypothesis is what is the chromatin accessibility landscape or DNA methylation landscape in these same cells? Do we see the binding sites for that transcription factor accessible in the same cells? Do we see downstream gene enhancers accessible in those same cells? So this is where single cell multiome technology comes in. So single cell multiome technologies jointly measure multiple biological modalities within the same cells. So providing not just one biological readout per molecular feature per cell, but 2 readouts per feature per cell, or possibly even more than 2, as we'll see much later in the presentation. So for the purposes of giving an example for the presentation today, I'll primarily focus on multiome technologies that jointly profile chromatin accessibility and gene expression. This is kind of the dominant subcategory of single cell multiome technologies. So I've highlighted mRNA and chromatin accessibility as the 2 biological modalities of interest here. So to sort of demonstrate at not too technical of a level how this technology might work, we all might be familiar with single cell RNA sequencing, which of course measures gene expression by targeting mature messenger RNAs for sequencing. The readout of a single cell RNA sequencing experiment is a gene expression count matrix with cells as rows and genes as columns, or flipped, a cell by gene matrix. Similarly, a unimodal or single biological modality single cell ATAC-seq experiment measures chromatin accessibility. It does this using an enzyme called a transposase enzyme, which targets regions of open chromatin specifically for sequencing. And the biological readout of a single-cell ATAC-seq experiment is a chromatin accessibility matrix, where the counts here represent how often a particular region of DNA was detected as accessible by the transposase enzyme. So here we have a cell by region matrix. This might be significantly higher dimensional than an RNA-seq readout. And I'll elaborate a bit later on what the molecular features, chromatin regions, might be specifically for this experiment. So single-cell RNA and ATAC-seq multiome data jointly measures RNA and ATAC across the same single cells. So both experiments are performed, and I'll give a zoom in on that in a second. And the ultimate readout is two feature matrices, a cell by gene matrix of expression and a cell by region matrix of chromatin accessibility. So ignoring the details in this figure, just to give kind of an intuition for how this works experimentally, this is an illustration of kind of the dominant technology in the multiome space, particularly in the RNA and ATAC multiome space. That is the 10x multiome technology. At a basic level, how this works is first, all cells in a sample are treated with the transposase enzyme, which again is the essential enzyme for producing the ATAC-seq readout. Second, using very advanced technology that definitely others might understand better than me, cell-specific and also molecule-specific barcodes are added to DNA and mRNA molecules within each cell in the sample. And finally, sequencing libraries are prepared. So at the end of the experiment, we have a lot of gene expression counts and also chromatin accessibility counts that are assigned to specific cells within your sample. All right, so hopefully that provides just enough technical detail to understand kind of the readouts and the potential downstream applications of these data. So I'll now move on to single-cell multiome analysis methods. So I'll first start with what questions can single-cell multiome data answer? So again, this field is kind of in its early stages. It's a very new technology. So instead of giving a super specific overview of one analysis method, I'm kind of going to give some broad descriptions of a few different areas of inquiry that have been explored so far. And again, as more data is generated, the types of questions we can answer with this data is only going to expand. So here's a few biological applications that I'm going to highlight. First, linking regulatory elements to their target genes and to their upstream transcription factors. Second, inferring developmental trajectories. Third, discovery of rare or transient cell types or states. And fourth, using one modality to predict another. So first, linking regulatory elements to their target genes. So this is perhaps one of the most established analyses of single-cell multiome RNA and ATAC data that's out there. As you can see, there's several methods that have already been published that perform this function. And the basic intuition behind this style of analysis is for a given candidate enhancer or ATAC region and candidate target gene, perhaps a gene in the cis region of the enhancer that we think might be targeted by the enhancer, we have from our single-cell multiome readout chromatin accessibility, the accessibility status of that enhancer across single cells and the expression of the candidate target gene across single cells. So at a broad level, what all of these methods do is measure the correlation or the association between the activity of a candidate enhancer and the expression of a candidate target gene across single cells, with, of course, some technical specifics that we might dive into a little bit later. But this is kind of the broad intuition of this type of analysis. And we can see how single-cell multiome data is uniquely unlocking this ability. We have high sample size in the form of thousands or tens of thousands of single cells across which to measure correlation. We can also really tailor the analysis specifically to our biological context of interest, which, because we know regulatory biology is very context specific, that's sort of a key benefit here. Another closely related application that has been explored is linking regulatory elements not only to their target genes, but also to the transcription factors that might interact with an enhancer to activate a target gene. So a few closely related methods like scenic plus and reunion highlighted down here might do this. And again, we're getting the gene expression, candidate target gene expression data from the RNA-seq. We're getting the enhancer data from chromatin accessibility. And we're getting the transcription factor activity data also from gene expression. So we can kind of measure the relationship of each of these things together as a network. A second broad application is inferring developmental trajectories. So there is this concept in regulatory biology, which many of us might be familiar with, called chromatin potential or lineage priming, which refers to an increase in accessibility of regulatory chromatin prior to a gene being turned on during development. So developmental biology is kind of this black box. There's a lot of, transient cell states and very context-specific biology going on that regulates the development of certain cell types, et cetera. And single-cell multiome data can be uniquely informative for this. So I'm going to highlight an example here from an early Multiome paper, the share-seq technology, which some of you might know, where on the bottom here, I'm showing that on the left side of the figure that will ultimately appear, we have the profile of gene expression and enhancer accessibility during early development. This particular example is for the development of hair follicles. On the right side of the figure that will ultimately appear, we have late development. And as the legend on the top says, red here indicates higher counts, so higher gene expression in this case, or higher chromatin accessibility in a second. But we're looking here at the profile of the Wnt3 gene, which, as we can see, becomes active late in the development of hair follicles. Down here, this bar that's being showed is gene expression currently. I'll now add one bar on top where actually the authors of this paper used the prior approaches to link regulatory elements to target genes. And now they're going to show the activity of those regulatory elements that were highly correlated to this target gene, Wnt3, throughout development of the hair follicle. So this new bar is showing the promoter of Wnt3, which, as we can see, becomes active, turns red a bit before the gene starts being expressed, which makes sense. We need the promoter to be active in order for all of the molecular cast of characters to come and express a gene. And interestingly, we also see that peaks that are correlated to Wnt3, which can be thought of as putative enhancers, each become active or accessible at some point in development prior to Wnt3 being expressed. So this example kind of shows how joint measurements of chromatin accessibility and gene expression across single cells can highlight this concept of chromatin potential or lineage priming in a way that is very difficult to access using other types of data. A third application is the discovery of rare or transient cell types or states. So this has been a major area of interest in regulatory biology as we have sort of discovered that regulatory relationships are very context specific. They might be specific to cell types, cell subtypes, or even specific cell states, like a cell being activated by a certain stimulus. We have become very interested in profiling these rare cell types that we might not even know exist. So dimension reduction and clustering algorithms, which I'll elaborate a bit more on later, are used to define cell types and states from unimodal data, very frequently from single-cell RNA-seq data and also from ATAC-seq data. But applying these methods to multimodal data increases our biological resolution and enables joint dimension reduction. So by using more features, more kind of unique identifiers of a particular biological profile, we can get better resolution on these cell types and states. And this can sort of unlock all of the other applications to be applied to specific contexts of interest, perhaps particularly relevant for disease or development. And a fourth kind of emerging application of single-cell multiome data is using one modality to predict another. So this example is from a paper that came out this year, which is cited a lot in this presentation. They did a lot in the paper, where one of their sort of problem formulations was to model gene expression as a function of cis chromatin accessibility and use those models to predict gene expression from chromatin accessibility alone. This is kind of an analytical framework that has been tried before in the bulk space, but of course, with lower resolution and lower sample size, it's challenging. So this paper really proposed this new area of inquiry that is uniquely enabled by single-cell multiome data. And we can see how this might actually have an interesting effect where it can help us unlock new insights from unimodal data. If we produce high-quality multimodal data, we can train models to predict one modality from another and then get double the insights from a unimodal experiment. For example, if we only did single-cell ATAC-seq and inferred gene expression from just the single-cell ATAC-seq. So big cost saver and biological insight increasing strategy. Okay, so those are just some of the broad types of questions that single-cell multiome data has been used to address so far. Certainly, there's many more on the horizon. Maybe one of particular interest to a medical and population genetics audience is as the number of data sets expands and even incorporates a large number of individuals, we can define very complex biological characteristics for genetic association studies. So for example, instead of measuring the association between a variant and the expression of gene A, perhaps we can measure the association of a variant and the correlation between an enhancer and a gene. Is there a particular variant that sort of unlocks a regulatory relationship? You can imagine a very multi-layered profile of biological characteristics that you could measure through genetic association studies. So for the second section of the analysis methods, I want to talk more about some of the technical challenges in single-cell multiome data analysis. So anyone kind of developing methods in this space or even applying methods in this space, single-cell data has unlocked a lot of potential, but it's also unlocked a lot of obstacles that we need to think through. A lot of other types of data don't really present us with this. So the first sort of challenge that I will profile is sparsity or noise in single-cell data. So the challenge here is that single-cell data is sparse, which means there are many zeros, for example, in a single-cell cell by feature count matrix, and also noisy, just due to the very small amount of biological material that's being measured. And the challenge here is that sparsity and noise is due to both true biological variation and incomplete sampling, what is sometimes referred to as dropout, where a gene may be truly expressed, for example, in a cell, but just because of the randomness of sampling, no reads show up in the cell. And it's particularly challenging because we cannot distinguish the biological absence of a feature from just the technical absence of a feature. So this is something that's very, very present on a lot of single-cell method development researchers' minds. And the reason this is challenging is because many traditional linear regression-based models assume normality of the data. So this plot down here kind of shows what the distribution of read counts, for example, for expression of some gene, might look like. There might be a big peak at 0 due to dropout and just kind of low profiling of a gene. And the frequencies continue to decrease as we move up in level of read count. So clearly this distribution does not look normal. And so a lot of traditional analysis methods might fail when applied to this data. So there's a few different solutions that are being actively sort of explored to address this challenge. First, I'll highlight some solutions that continue to, at their core, use a normal model to model single-cell data, but they just make some adjustments to the data to make the data fit the normal model better. The first is aggregating single cells into metacells. So single cells provide a tiny amount of biological material, and their measurements are, as a result, very noisy. But what some people are doing now is using dimension reduction algorithms and clustering, defining what are called metacells, which are sort of low-level aggregates of cells. So a few cells that are very close to each other in low-dimensional space and are therefore very biologically similar. We can group them together, aggregate their information, and get, hopefully, sort of a denoised but still accurate profile of the biology of those cells. So this might look like, again, grouping a cluster of cells and simply adding read counts. And we can imagine, specifically with respect to the dropout challenge, if each cell kind of has a random subset of molecular features, maybe genes that have dropped out, then hopefully if we aggregate across different cells, we have some information for all features and we're less prone to that dropout. So there's a few methods here for aggregating single cells into metacells. Another strategy that's been explored is imputing missing counts from single cell data. So for example, the MAGIC method used, again, this unique opportunity of having jointly profiled modalities to predict one modality from another and impute missing data to sort of smooth out the noise and missingness in this data. So very well-established approach in other areas of biology and can also be applied to single cell data. And again, retains the dimensionality of the data. We're still looking at a single cell level, whereas with metacells, we've reduced the dimensionality. We're now looking at a lower number of cells.
I have a question about that.
Yes.
Could you sort of talk about, you've mentioned two solutions, about when would you want to use one versus the other? What are sort of the limitations?
Yes. So I think in terms of the technical solutions to address some of the challenges in single cell data, interestingly, they have not been rigorously benchmarked against each other in a lot of cases. A lot of times, many people are developing methods simultaneously and they will sort of propose an approach and it's reasonable, it's been used before, but we don't necessarily know the specifics of the approach, how they compare to others. So I can't say that I myself am familiar with sort of a rigorous comparison of imputing missing counts versus aggregating single cells into metacells. I think this could depend on your biological question. It could just be a matter of practicality. If you have some analysis procedure that's very computationally costly and it's beneficial to reduce the dimensions of your data, perhaps a good denoising strategy is to aggregate single cells into metacells, which will do both of those things. But I think in general, both of these are kind of accepted strategies in the field. So it really depends on your particular data set, your particular question of interest. Another consideration might be just how specific is the biology you're looking for. If you really don't need to look beyond the level of a cell subtype or a little cell village, then aggregating single cells into metacells might be a great idea because you're not sort of introducing new data. However, if you're really interested in the biology of single cells, maybe you want to retain the dimensionality and impute missing counts instead, if you don't mind having some predicted data in there, not just raw data. So those could be a few considerations when thinking of those two. Yeah. All right, so yet a third approach, and perhaps the most basic, maybe the first approaches that were tried, is normalizing or transforming read counts within single cells just to look more normal. And this figure might not really communicate this, but this is pretty challenging given just the sheer level of sparsity and noise in single cell data. So sometimes this is not enough, and the first two approaches might be better, or some combination of the three. So a second category of solutions is using a non-normal model. And this is certainly an area that's been explored a lot recently. So discrete count models, like Poisson models or negative binomial models, might be a more appropriate fit for single cell data at the single cell resolution. A couple of methods are highlighted here that use those approaches. And beyond just the vanilla Poisson or negative binomial models, we can also consider zero inflated models that explicitly account for the fact that there's going to be a greater than expected amount of zeros. This, again, is an area of active development, and I'm not aware of anyone who has rigorously shown that one model, zero inflated or not zero inflated, is sort of better than the other. It's, again, this space where a lot of people have a lot of ideas, and they're all honestly acceptable right now. So it could be a matter of what fits your data best and what fits your biological question best. Okay, so a second sort of half challenge, half feature of the data is high dimensionality. So as we've been saying, single cell data is highly dimensional. There is number of features times number of cells, number of data points, which is vastly larger than bulk sequencing data sets. And while this is a key feature of single cell data in the sense that it gives us high sample size and often very high power in a lot of analyses, it also might increase computational complexity and hinder interpretability in some other contexts. For example, if we really want to make inference at the level of cell subtypes or cell subpopulations, having a bunch of single cells might actually kind of overwhelm us and not really provide a lot of easily interpretable results. So this is just demonstrating how the dimensionality of data increases as we move from bulk to single cell unimodal data, and then even further as we go to single cell multimodal data. So some solutions to this in a setting where this is viewed as a problem is reducing the number of features via feature selection. So in the RNA space, this might look like restricting only to highly variably expressed genes. In the attack space, this might look like calling attack peaks, identifying regions of the genome that are particularly highly active, or defining genomic tiles, kind of partitioning the genome into discrete sections that we're going to aggregate chromatin accessibility across, rather than measuring chromatin accessibility at the base pair level. And a second thing we can do to reduce the number of, perhaps let's just say number of entries in the data matrix, is dimension reduction on cells. So this is a very well-known kind of area of inquiry using PCA or NMF, perhaps combined with some other nonlinear dimension reduction technique to reduce the dimensions of the data. Or I suppose that also reduces the features, not that, okay, yes, my bad. That also reduces the number of features. Next, there are strategies you can use to reduce the sample size or the number of units of analysis. And again, I've provided here the strategy of aggregating single cells into metacells, forming those sort of cell clusters of closely related biological neighbors. So obviously, this is kind of a big landscape of solutions that I've provided to these two big challenges or features of single cell data. And it's kind of, as I said in response to the question, a uniquely tailored solution depending on the biological problem of interest. What dimensionality of data can your analysis withstand? And what is going to give you highest power and greatest interpretability? So these are just kind of a menu of solutions available to people doing single cell data analysis. Okay, so lastly, to kind of bring together the broad types of questions that single cell multiome data can answer with the technical challenges, I'll highlight a particular area of inquiry, linking enhancers to genes, to sort of highlight the unique benefits of single cell multiome data. So I'm sure I don't have to convince any of you that mapping gene regulatory architectures is critical to understanding disease. The vast majority of disease-associated variants in GWAS are non-coding, and these variants often do not regulate the nearest gene. So linking regulatory variants to their target genes is a critical step in interpreting GWAS discoveries and nominating potential drug targets. And historically, regulatory activity relationships between enhancers and genes have been interrogated using various lines of biological evidence. Most recently, or one of the newer developments on the scene is, of course, single-cell molecular profiling, specifically single-cell multimodal profiling. And compared to some of these other methods, single-cell multiome data is context-specific. So information we might get from DNA sequence or conservation predictions really doesn't tell us any information that's specific to biological contexts. And as I said, we know that regulatory biology is very context specific. So that's a point for single cell multiome data here. Single cell multiome data is also closer to causality than some genome-wide biochemical assays. For example, 3D conformation mapping and biochemical annotations like DNA methylation or histone marks. These provide genome-wide kind of predictions and proxies of regulatory activity, but they don't really hint at causal relationships. And sometimes they don't even implicate a target gene for a candidate enhancer. Whereas the correlation between an enhancer and a gene is a closer proxy for biological causality. Single-cell multiome data is easier to collect. So relative to something like EQTL, where we need to collect hundreds or thousands of bulk sequencing samples to run a genetic association study and identify relationships between regulatory regions and, for example, gene expression. Single-cell multiome data, we can often derive a lot of biological insights from a single sample, from a single donor even. Single-cell multiome data is also genome-wide and convenient. So relative to more targeted experimental approaches that often seek to address a specific biological hypothesis about one gene or one enhancer, single-cell multiome data provides genome-wide predictions and is not nearly as experimentally burdensome as profiling many genes through a CRISPR screen, for example. So as I briefly highlighted before, single-cell multiome data has been used to link regulatory elements to their target genes by measuring the correlation between the accessibility of a region of chromatin, a candidate enhancer, and the expression of a candidate target gene. And if we repeat these pairwise tests for all enhancer gene pairs, we can sort of predict or map regulatory biology genome-wide using a single sample. And so in this table, I've highlighted a few specific analysis methods just to demonstrate how different strategies are being employed to address the challenges that we discussed in the previous section. So for example, the scent and scar link methods for linking enhancers to genes perform analyses at the level of a single cell, but to address the non-normality in the data, they both employ Poisson regression instead of measuring linear correlation or using linear regression. A third method, the Signac peak gene linking method, also performs analysis at the level of single cells and does measure linear correlations, but performs specific normalization steps to make the data look as normal as possible and make the correlation readouts as meaningful as possible. A fourth method, the Archer method, measures simple linear correlation, which assumes normality of the data, but importantly, it aggregates single cells into metacells using a joint dimensionality reduction prior to measuring correlation. All right, so this is just, again, a specific example of how these different challenges and opportunities from single cell can be combined to design strategies to learn about context-specific biology. So a few conclusions. Single-cell multiome technologies jointly profile multiple biological modalities while retaining cellular heterogeneity. This is kind of the key benefit of single-cell multiome data. Single-cell multiome analyses can target diverse biological questions, some of which I've highlighted here, but many of which will be unlocked in the coming years as more data is generated. Single-cell multiome methods use various strategies to handle data sparsity or noise and high dimensionality in single-cell data. And finally, we took a somewhat closer look at several computational methods that use single-cell RNA and attack multiome data to link regulatory elements to their target genes. So one final point, a bit of a forecast, is that these multiome technologies and analysis methods are rapidly developing. So for example, we highlighted kind of the dominant technology today, which is RNA plus attack data, but another technology that jointly profiles RNA, gene expression, and cell surface protein is also kind of rising in popularity. And we can imagine how that would unlock unique insights as well. Some overlap with the applications of RNA plus attack, but some new biological questions. And there are even emerging technologies that measure 3 modalities at the same time. For example, gene expression, chromatin accessibility, and DNA methylation, or gene expression, chromatin accessibility, and intranuclear proteins. So we can imagine the number of biological questions just multiplies as we can combine multiple modalities within the same analysis unit, same single cell. And the vast array of the technologies that are available are reviewed thoroughly in this review that I've highlighted here, Van der Eycken et al. 2023, Nature Reviews Genetics. All right, so that is all I had for today. I'm happy to take any questions. Thank you very much.
Thanks, and if you guys have questions, just come up to the microphones, and while we wait for them, I'm wondering if you could touch on quality control. Is it as simple as doing each modality separate, and are some things that you consider jointly?
Good question. So quality control is, because of the sparsity of noise in single cell data, a huge consideration. There are sort of separate quality control, kind of established quality control procedures for RNA-seq data and attack-seq data at the single cell level. And multimodal quality control is pretty much just the intersection of those two, which is challenging in the sense that if you think of a subset of your large single cell sample being of high quality with respect to RNA, and a subset of that data also being high quality with respect to attack, sometimes those will be correlated and sometimes they will not. So finding the intersection of high quality cells for both modalities is often a challenge and often results in somewhat smaller data sets, fewer cells than one modality alone. But it's mostly just a function of performing both types of QC.
Are there any methods that maybe if a cell in the RNA-seq is like borderline, and then you look in the attack-seq and it looks great, then you might be like, keep it, versus in the attacks, like, does that exist?
That's a good question. So to my knowledge, it's pretty much user controlled. When you perform your QC, you can sort of decide what your cutoffs are going to be for the number of reads of attack per cell and things of that nature. So I would imagine there is a way to sort of ad hoc define some quality score per cell per modality and cut off at like a product of quality scores between the two modalities or something like that. Yeah, that would probably be a good strategy to increase the ultimate dimensionality of the data.
While the audience is gathering their questions, where do you, if someone is new to single cell and multiome data, where's a great place to look for data sets to get their hands wet?
That's a great question. So 10x Genomics has several multiome data sets that are publicly available that are a good place to start. They're often already processed, already in kind of feature matrices for you. And a lot of papers are coming out these days where they have generated single cell multiome data sets in specific biological contexts of interest or using a new technology, and they will often also release their data. So I would say 10x Genomics is a great place to look, and also just kind of keyword searching single cell multiome data in your journal of choice, maybe the Nature Journals family or something like that. And you can often find some good data sets that way. And often, sorry, before you ask your question, I would say another recommendation is any paper that is proposing a single cell data analysis method really for any part of the process, whether it's kind of a data processing method or a downstream kind of biological question. You can also look at the data sets they have used, because oftentimes those authors have gone through the process of picking out these data sets from corners of the scientific world to analyze. So those would be my three recommendations.
If you have different modes of measurement or like different modalities, can you use the same software to do this, the analysis? Like, as long as you have some sort of cell by feature matrix, can you process everything the same way?
Good question. So it really depends. And I think the answer is there's not currently kind of a unified approach that everyone in the single cell world is using to process their data, because the data, the technology is just so new. Everyone's kind of doing their own thing to some extent. The key kind of consideration to keep in mind is the differences between RNA-seq data and ATAC-seq data. They are similar in the sense that you're getting kind of read counts for both. I would say the main difference is ATAC-seq data is much higher dimensional, and each molecular feature is going to have higher variance and not be as well profiled. So with that principle in mind, there will be probably some differences in how you process the data. But usually the same software can be used for both. So for example, Surat and Signac are kind of major softwares in R. Also, Archer is a big software more tailored to single cell ATAC-seq, but also developing applications for integrating RNA-seq as well, sort of in multimodal data. But typically, I think you should aim to use the same software package and then integrate them using that software package. Usually they have that functionality. That's a good question.
Thanks so much for your talk. One of the really subtle things you mentioned was the opportunity to look at the impact of gene variants on the correlation between enhancers and gene expression. Mm-hmm. Can you say any more about how people do that and the challenges of doing that and the challenges of having enough data to do that?
Yes. Or a description of how it's been done that was kind of just invented in my mind. I think for unimodal single cell data, this is obviously an area of hugely expanding interest, measuring the association between genetic variation and chromatin accessibility at a known enhancer or methylation at a known site or something like that. So I think as we combine biological modalities together, the space of possible phenotypes that we can measure genetic associations with just expands. And as you said, the big challenge with this is, are we ever going to have data sets where we have hundreds or thousands of individuals with genetic data and multiome data? Because multiome data at the moment is quite costly and burdensome to produce. So some of the more well-established lines of inquiry really only rely on data from a single sample, like linking enhancers to genes. If we got to a place where we had some atlas of hundreds of individuals with genetic variation and multimodal measurements, I think it would be incredibly powerful to be able to perform association studies on very complex phenotypes like that. The specific example you highlighted, in my mind, perhaps it could reveal some sort of context specificity or stimulus dependence of a regulatory relationship. For example, if people with a certain variant exhibit a strong correlation between an enhancer and a gene and people with the alternate allele do not, what does that say about the function of that variant? Do we know that it is in a transcription factor binding site or in an enhancer for another gene that's closely related? Like what information might that give us about the interactions between these different biological processes? So that was just kind of off the cuff. I wish I had a specific study to cite, but maybe in the future.